kind of these diagonal lines going, I guess that would be, yeah, like,
That?
yeah, we're sort of curious.
Yep.
Is that an artifact of how we collect bathymetry?
It's an artifact of multi-beam bathymetry. So what you're seeing are individual pings, right? And so it looks really ragged out there because that's the outer edge of the swath. So in the outer edge of the swath, you're getting more kind of bending up or down of the beams just because it's further for it to go. And so it will be a bit bumped out a bit higher. And if, depending on the gridding resolution, sometimes if we grid it, we could grid it at a lower resolution and it would smooth out a bit of that.
Okay.
But we kind of push the resolution elsewhere, so that becomes more prevalent, if that makes sense. So yeah, each of those, like if you could picture the multi-beam clicking along at, you know, a 45 kind of this angle, that's our track line, and each of those little lines is just an individual ping that's we're showing up in the bathymetry. Cool. You can see it down here as well. It's just really prevalent up there. You'll get more of that often as we go deeper, because you're going to have a little more uncertainty in the outer edges of the beam.
Cool. Thank you. That's very helpful.
Yep.
So to do the little bit of mapping that we did last night to get some really fresh data for this site, like what would be the length of the lines and the space between the lines that the ship would do?
Length of the line would just depend on how big the site is and how much we want to cover. And then the spacing, we typically try to overlap our lines 100% so that the edge of one swath meets the center of the other swath. And that helps to negate some of that outer edge noise because you're getting the data directly under the ship at nadir. The sound has less of a travel, right? So you're getting better data there and in the inner beams. So if we can overlap a lot, it helps kind of smooth out the noise in the outer beams. So depending on water depth, the like hardest or loose estimate is like 3 to 4 times water depth is the width of your swath. It will depend. As we get past 3,000 meters, that tends to come in a bit. Sometimes we're down as low as 1 to 2 times water depth is the width of our swath. So we plan based on maybe 2 to 3 times water depth, matching the edge to the center, and then we watch it. And if we're not getting enough overlap, we might move the ship in a bit to get better spacing and better overlap.
Okay. Excellent. Thank you so much for explaining that.
Oh, anytime.
We don't get to talk about the mapping quite as much, but it's such an important part of planning and executing this type of work, especially when it entails a lot of exploration in regions people haven't been. And Lisa and I and the rest of the science team have been just going gaga over how wonderful it is to have Nautilus and the OET's team helping us do all the mapping and make these decisions. We can spend hours debating on what's the best terrain and having these wonderful maps. And they never complain when we ask them to remap something or show it a different way or spin the 3D thing a million times.
Oh, we love it. The other thing we can kind of do, I mean, the resolution of the data we can get is just limited by physics, more or less. Like the ship is on the surface, the multi-beam is a certain frequency, so you can only get a certain level of resolution on that. But what some games we do try to play is like really slowing the ship up. So sometimes we can map as slow as like 3 or 4 knots just to try and get more measurements in tighter spacing along the seafloor and improve the resolution a bit. I think Lindsay was playing some of those games last night to see what he could do.
Yeah, I'd like to second Kira's comment about the fantastic mapping support. It's really made our life much, much easier. And the responsiveness has also been great. Basically, any time of the night or day, we have a mapper who can help us look at things, help project the images up to our nightly science meetings to help everybody else understand what's going on. And then literally an hour before the dive, we can look at the latest processed mapping data and make tweaks and changes to the dive track to try to optimize our sampling efforts. So it's really been fantastic. Thank you.
Well, thanks, Lisa. That's very kind of you to say. We love doing it.
Yeah, and if we really want to keep nerding out on the maps while we continue to go on our descent, we're currently at about 1,300 meters, and we're shooting for 1770. So for those watching in, you've got about 400 more meters of marine snow to catch as the Herc camera flies by.
14 minutes.
Record speed, 14 minutes to go.
Lightning speed to the seafloor.
400 meters. And Mark Legg, one of the geologists chiming in, is telling us that near the top of the escarpment in our map that hopefully you viewers at home can see, there is a narrow linear flat area that is actually a fault bench and probably an active fault. So perhaps we can see some interesting live footage if we can get up that high during our dive.
Is he talking one of these intermediate ledges or up in here somewhere, do you know?
Yeah, Mark, if you can chime in. I think, can they see this map on the quad view?
They can see HIPAC, yeah.
Yes, okay, cool. Thank you. Yeah, we're curious if, Mark, you can point out which ledge is the most prominent for that fault feature.
Probably hard for him to describe. It's hard to see the waypoints, but that's AP there.
And just like Lisa mentioned a little while ago, we're now getting into the more oxygenated bottom water. We're at about 7% oxygen saturation at around 1,400 meters we're coming up to. And just around 630 meters, we are at 1% oxygen. So now we're getting into the higher saturated.
What about temperature, or the current temperature in the water?
Maybe
3.2.
3.2,
3.2 Celsius.
pretty cold.
Pretty cold.
Oh, they're broadcasting the back view today. So make sure you look, the back of your head looks cool if you want to wave.
That's what's being broadcast. Back of our heads.
Well, they get to see the amazing monitor displays that we get to see. It's funny, this is my first, I've been on a cruise where we did some dredging and some sampling in San Francisco Bay. And this is my first ROV cruise, and I thought that it would be difficult, or that the time would pass so slowly if you were watching the seafloor for 4 hours, but it always goes so fast. It's super fascinating. Everybody told me it would be like that, but now I've experienced it for myself, and even when my shift is over, I find myself in the lounge just staring, hoping to see the next cool animal or the next best rock. And usually you guys are doing 24-hour operations in the ROV. What would be your typical schedule in a non-COVID crew?
We're sailing short this cruise. We only have 4 people in the ROV group. We normally have 6. So we have, 2 people per watch. We do a 4-hour on, 8-hour off cycle.
And so the ROV can stay down.
It can stay down for 3 or 4 days, really.
Wow.
And we often do. That's more typical operations.
We usually have a 4-hour turnaround from on deck to back in the water.
And so why do you bring it on deck?
You gotta check all the
Samples.
systems and unload samples.
Yeah.
Oh yeah.
Get full of
Samples,
samples.
that's right.
Yeah, we check the oil and fill up the tank.
Yeah, I brought one really large cooler for rocks and I've almost filled it up. I'm not going to be able to lift it into my car after this.
We'll have to crane your cooler
That's
into your
one
car.
good thing about coming up so regularly is we're always bringing up a lot of samples.
Well, I once had a cruise. We only had 6 days to collect a lot of experiments on the seafloor using Jason. And we put Jason down on the first day and it stayed down for 6 days. And we used gear elevators to bring samples up. We had 2 gear elevators on the ship and they rotated on a 12-hour cycle. So every 12 hours we had an elevator full of samples coming up with Jason staying down on the bottom.
Wow.
Is Jason another ROV?
Jason is an ROV, yep, in the UNOL system. So that was a 6-day Jason dive, 1 launch and 1 recovery.
I was a pilot of both Jason 1 and Jason 2.
Cool.
Years. Yeah.
But were you on that dive?
Maybe. When was it?
Ooh, a long time ago.
Yeah.
You would have
I did it
remembered
for-
Bob on there.
Yeah.
I want to say it was somewhere.
I think I've been in a submarine with Lisa.
Maybe
So.
maybe 2006 or 2000 somewhere.
Yeah.
2008.
That sounds epic.
That was really hard work for the scientists because samples were coming up all the time. As soon as you finish your watch inside the Jason van, you had to go out and get the elevator ready and process samples, get the next elevator ready. But it was an effective use of the.
Yeah. What was the depth? I don't, how?
It wasn't deep. It was in Oregon at methane seep. So the depths were 600 to 800 meters.
Mm.
Oh, Greg said it was Hydrate Ridge in 2011. I guess I got my dates wrong.
Yeah.
Thank you, Greg.
Oh, we're back up to 9% oxygen saturation.
Yeah, some of our higher. Trying to remember what it was yesterday at the day before yesterday at the 500 some meter depths. It's high also, a little higher.
So
We're at 40 micromolar, so that's considerably higher. We're not in the oxygen minimum zone technically anymore. Well, corrected, it's 33 micromolar. We consider below 20 to be the oxygen minimum zone.
You set that to the right gain? Yep.
yeah, that's it. To the southeast. Well, I'll look forward to this.
So we're at 1,600 meters. We're getting there.
That's encouraging. Mark Legg says that they recovered a very large manganese nodule in quotes, which sounds about right since it was about the size of a small suitcase from the base of the linear ridge in South San Clemente.
Oh, cool.
So hopefully I was right in my guess that we'll find some ferromanganese coated stuff down here.
So one of the things that I didn't really mention in terms of the motivation for this cruise is that ferromanganese crusts are being targeted for minerals mining in the seabed in a variety of places, in international waters on seamounts in the West Pacific in particular, and I think the Rio Grande Rise.
Rio Grande Rise in the Atlantic.
Yeah, off Brazil. So there is no specific mining interest here in Southern California, but we're hoping to learn more about the ecosystems associated with those crusts by studying them here.
Yeah, and for crusts specifically, most of those regions are still in the exploration phase. They're definitely being considered for their resource potential. Mining itself is pretty far off for these deposits since they coat the rocks. It would be very difficult to extract them, and that technology doesn't exist yet. But certainly mineral resource potential of these ferromanganese crusts is a hot topic. And so determining the distribution in this region and the associated biology and the ecosystem resources that are part of this potential mineral resource is a very useful and important field of study.
And we haven't really been talking much about what we've been finding on all the rocks that we've collected, but for people who are interested, there's a really odd assortment of small organisms. Some of them are protozoa, agglutinated foraminifera, for example. There's usually some small sponges, occasionally a limpet.
Those ophiuroids.
Polychaetes, and yes, there's an
My
ophiuroid.
favorite.
Devon's favorite little creature. You want to describe that one?
Yeah, it's incredibly tiny. So when we're processing our rock, we need a headlamp to see the little sheen on it. And then it's less than a millimeter across, and it's so incredibly tiny, but it's really satisfying to pick those guys off of that rock.
So ophiuroids are brittle stars, which are related to starfish. And this particular ophiuroid that we have found on almost every rock we've collected has itty bitty arms. They almost look vestigial, like it's evolved away its arms, which makes me think this is a sessile ophiuroid and not one that crawls around the seafloor. So we've seen a lot of big orange ones with spiky spines on their arms, but this rock ophiuroid is mostly disc, the central disc with arms that are maybe only, I don't know, a fifth or a tenth the length of the disc diameter. So little bitty arms.
Are you guys photographing things you see under the microscope?
Greg is.
Greg is. Yeah.
Okay, I'm gonna have to check some of it out. It sounds really cool.
Yeah.
Yeah, Greg, I'm really hoping you've got a picture of the ophiuroid with the tiny arms. That's one of maybe not the poster child, but it is probably the most common animal that we've found on the rocks.
A lot of those gromids too.
And gromids.
Little sausage link type thing.
Which are single cell protists.
Oh, okay. I know. I remember seeing those guys.
They look like little grapes hanging onto the rock.
Yep.
Sometimes zoom in on them, and they're interesting. They have an clear organic wall, and inside it really just looks like sediment. I'm pretty sure that's their fecal material that's being stored to create structure.
And
I'll stop.
The common name for the ophiuroid that you were talking about is brittle star, right?
Yes.
Okay.
And Greg has confirmed that he's got some good photographs of both the brittle stars and the gromids, and he's going to create a best of set for us from this cruise.
Oh,
Best
cool.
of
Little
pics.
highlight reel.
Greg's photographs are amazing. You know, keep trying to get him to make a coffee table book of deep
Yeah,
sea
that would
pictures.
be awesome.
All right,
Yeah.
we're getting close to bottom.
You can go auto ahead.
Sure. Right.
Ooh, we're getting there.
And we're going to want to take a Niskin before we core, right?
That's right.
Thanks for the reminder. Totally forgot.
Good catch. So do you think it's colder in here because we're diving deeper?
Feels like it.
Yeah. It's part of the experience.
It's 2.7 degrees now.
Okay, Kylie, let's start coming down together.
Sure.
I think I got a visual.
There's more brittle star on the bottom. Looks like it.
Lots of them.
Oh yeah.
Lots of little stars.
Oh, they look like little sparkles for a second.
Yeah.
I see them now.
It's like starry, starry night.
And these look more red, or more white, sorry, than
Yeah.
the red ones
Not
we've
the
been
typical
seeing.
orange ones we've been seeing.
Different species.
You want to do your do like you do?
Yep. I will do what I do. Are you gonna Niskin?
Yes, a Niskin, please. Whenever you're ready. No rush.
And this will be sample NA12446 Niskin 1.
Okay, am I okay to go?
Yep.
All right. Okay, you're reset.
And are we 4.9 meters off bottom, Aaron?
Yes.
Yes.
All right.
You want to be closer? Or?
This is fine.
Higher. You got it.
Watching?
Yeah. Oh, yeah, you pulled it.
Was that 167?
Yes, 167.
Okay.
$17.73.
Yeah.
And for those of watching that we said we pulled a Niskin, that's a sample type for sampling seawater at the depth of the Herc vehicle. When they pull that little cord, it opens up a little valve and water from this depth rushes into the chamber and you get a capture of the seawater for this depth and location. And I think the biology team will be analyzing those for carbon and some DNA to determine the background concentrations and biological community to compare with their cores.
Yep, and we also take a sample at the top, so then we compare the surface water to the temperature, or the carbon we take at the bottom.
Awesome.
And those are the Niskin bottles on channel 3 on the quad.
You guys got the position on that?
Yes.
Okay.
So are we sampling here?
Yes, we'd like to take 8 push cores, please.
The sea stars look like
There's a looks like there's a brisingid out there.
Any ID on this brittle star? Are they the same species, just a different color, or?
I think they look like Enresia.
No,
No?
that's not a brittle star. Sorry. Enresia is an asteroid, but these are brittle stars, ophiuroids, which is a different group.
Got it.
So I was going to guess Ophiumusum, and that's a possibility.
Again, how come I was getting yanked?
It's hard to hear you, Bob.
How come I was getting yanked just then?
Pretty stretched out.
Sorry, I need to get a new spot there. I was getting yanked around a bit. I
Okay.
don't know, just come down.
I am. I gotcha. I'll give you a leash.
Is that another Panicia?
Maybe. I'll have to take a closer look.
Hard to tell.
How's that? More?
Good.
Okay.
These are good sized brittle stars,
Yeah.
more than 10 centimeters across, some may be 20. Probably well fed, high densities. The more animals you see, the more food there is. Sort of the rule of
Any
thumb.
particular spot you want to land here?
No,
Nope.
just the core should not be over a brittle star,
Yeah.
preferably. It's very fine
we,
sediment.
Can you move up to me a little bit?
Yeah,
I
no
think I'm
worries.
getting
What are we
jerked
at,
around.
315?
Yeah.
318? Bridge nav. Can we get a 20 meter step bearing 318? Thank you.
And Kylie, you can come down some more. We're stretched out.
Yep, no
Don't wanna-
problem.
I don't want to stir up the bottom more than I already have.
Okay.
You're just gonna have to come up when
Actually-
you get closer.
Okay.
I think we're back in the depths of not believing this very much. Just FYI.
Oh, but I
So-
can believe that we're far away.
Yeah, no, I'm coming in on your heading, if that's all right.
Yeah.
That's what I'm going to go with.
Yeah. 1700 shouldn't be too bad. All right, I'm just gonna land. I'm just gonna deal with it. Oh, look at all these critters, though.
Chips coming in.
Bob, I'm going to change my mind and just ask for 5 here.
Okay.
And the reason is that there's possibly seep type habitat up ahead, and I want to save some cores for that in
Okay.
Roger that.
case we get to find that.
Yeah, Mark Legg, the geologist in the chat, is saying that the first scarp we're coming up to looks like a restraining bend in the main fault zone. And restraining bends are notorious for seeps.
Ooh, I like the sound of that.
No guarantees, but
Yeah, we got our explore mode on.
It's nice having all these geologists in the chat. Makes things easy for us.
we have experts in the region, which is
Yeah.
it's really special.
Yeah. I can certainly do the minerals and some
But-
geology, but these guys are the real.
But I do have to say that even last night or this morning in planning our dive, I did comment to Lindsay that I hope that first ridge was seep carbonate because seeps are notorious for being at the base of escarpments like this.
We love seep hunting on the Nautilus. We had a lot of fun on Andrew's cruise, rolling through the multi-beam.
Switch over to sample mode.
Sure.
So we have a request for the warty sea cucumber. I guess there's one up ahead after you're done coring.
All right.
Zoom in on Argus.
Tore out.
Yep. Sample tray out.
It's like
Okay.
sample overload.
Actually, can you come wide on Argus? I expect the ship to move, so I want to be able to tell where I am. Thank you.
You do.
The ship's within 5 meters, the end of its move. Do you want another one in?
No, I just
Yeah,
wanted.
yeah, you can give me another one.
Okay. I was just nervous.
It's good for me though that I can see that view. That I can.
All right. Well,
that makes it faster for me because I can see.
Yeah, if we're at the end of the move, no problem.
Okay.
I'm not usually this low. I just wanted to.
Yeah, well, you're gonna have to. Yeah, you're gonna have to come up when we.
It's gonna swing in a bit too.
Okay.
So I'll let it settle out, and then if we need more, just let me know.
Okay. You want dive mode?
Yeah. Where am I gonna put it? I'm gonna put it right there. Zoom in. Yeah. Zoom in.
That's So for folks joining in and watching, these are called tube cores. Those are empty plastic tubes that we sink into the sediment, hoping to preserve the full column of sediment inside the tube from the surface down to as far as Bob can shove them in there. And when we lift them up, they hopefully stay in the tube and we store them in the quiver. And when they come up,
Lydia,
the
go ahead and zoom on Argus.
biology teams will sample them at different depth intervals to look at the communities of biological organisms throughout different depths in the core.
Erin, how close are we to the original bottom target?
We are, stand by. 7 meters.
Zoom in.
Great. Okay, we're going to want to make sure when we head, when we leave here, that we try to replicate our dive track as much as possible.
Okay. Do you want to make sure we cover that spot directly, the first point?
Well, possibly just to make sure we go over the highest point on the first ridge of rocks.
Okay.
It looks like those brittle stars are everywhere.
There's definitely a lot of brittle stars.
Some are covered with sediment.
This is typical of the basin areas in the borderland. The Santa Catalina Basin's covered with brittle stars. As long as the basin's not anoxic, the brittle stars are there. Basins are like food traps for them.
Yeah, wow, we're at 37 micromolar oxygen here. Another good reason to go deeper for ferromanganese crusts hunting, other than the lower sedimentation rates, is that crusts can be thick and particularly high in manganese just below an oxygen minimum zone, because in the low oxygen waters, manganese is dissolved and goes into the dissolved form. And so when it hits these more oxygenated waters, a lot of manganese precipitates. So hopefully we'll see that happening along the cliffs as we climb towards the OMZ, oxygen minimum zone.
Look, there's a star right under the surface there.
Yeah. Over here.
They're everywhere.
Sorry. I like this little zigzag pattern we're getting. That looks like pretty fine sediment.
It's very fine.
Very fine.
You guys will be washing mud out of your core sampler.
So fine has two meanings here. Fine means we're happy with it, and fine means the particle sizes are small.
Yeah.
You switch over.
Is that a jelly or tunicate we're seeing in Argus's view, Lisa? Bottom of the screen.
Oh, it just left. I didn't see it.
That's right, I got some pictures of it.
Yeah, I'll go over here. Starfish free?
I can't see any.
Maybe.
Right.
Ooh,
Oh.
look at that beautiful polychaete
That was cool.
scampering across our field. Okay, is that what you were talking about?
Yeah.
I didn't get a close look at it. So I mean, it could be a sea cucumber. I didn't. Is it the same one?
Yeah.
It's coming and going?
Yep.
What's the sample number?
This is NA12446.
That's core
Which
5.
Core 5.
Push core 5, yes.
Oh.
Oh no. I typed too
Yeah,
soon. I
that was
jinxed it.
a botch.
I had it all set up for you, Al.
I'm missing something. It's out of frame now.
It was in frame.
I can't change pan and tilt. I can only zoom.
I know. I'm setting it up for you. When you zoomed out, it was in.
Quite the awkward angle.
Boy.
Okay. It had changed. Okay. No, no, your fault. It ran away. Let's yell at it when it gets back. No grog. Right.
What's happening?
Core withdrawal.
Oh.
It's lying across.
Oh, I see. I looked down. I was taking notes, and I looked down. And
Would it help to have box in a little?
No. Box out, maybe.
box out. Coming out. They went okay.
Yep, right there.
Oops, sorry. Want to bump in?
A slight tilt down on Argus so I can give Bob a better view. If you have it in the left.
Yeah, that's a backwards view, though.
Oh.
That's about all I got for down.
Dang it. All right, bring it in some.
Okay, coming in. We'll tray in.
Keep coming. Okay. Boy. Be one of those days.
That's too far aft. Come tight on Argus, Al.
So you want to come out wider?
Come tight on the box, and I'll see if I can help him with the.
It's gonna be tough. You get tilted all the way down too, aren't you?
Yeah.
Stuck back there good.
So frustrating.
Aft a tiny bit. There you go. That's.
Stay.
Oh, that's precarious.
Yeah. Back where it was.
Aft a little. You're.
Um,
Keep going aft. Yep. Now close. Yep. You got it.
Is that still viable?
It seems like it's still got sample.
Yeah, but is it all?
you want to shake it out?
Yes, yes, please.
You're too far. I can't hear you.
If you can shake it out away from the other tube cores, that would really be helpful.
Out here?
And if it doesn't come out, we'll just sacrifice that core and put it back in so we don't spend too much more time here.
You switch me around?
Yeah, sure. Sorry.
It's like things are still falling out. Yep. Maybe not, it's not coming out much. Let's see. You think that's just dust inside, sort of sediment now? If you turn it upside down. And, yeah.
Doesn't look like sediment. Seems like
Just suspended material, right? Would you guys be able to use that core or not, Gabe?
Yeah, I think if we retry it, yeah, we'll.
But with that suspended sediment in there?
Yeah, I think it'll be best not to have it, but yeah, since we're really
It looks
focusing on that top portion, that's kind of like the best sample, I think, for us.
Okay, maybe you can just punch that one in and pull it again, and maybe the biologists will use it.
Well, it's pretty clear.
It's
I mean,
pretty good. It's pretty
yeah.
good.
Yeah, that looks good.
I got some photos of it, and then we could just add that in the notes.
I think it's fine now.
Yeah.
Okay.
Caitlin's saying it looks fine.
Yep.
Just to record it.
And thank you, Bob, she says.
Okay. Can we zoom in? That good?
Yep.
I can't tell if it's starfish free.
It is.
All right. One more time.
Aft a bit more. That's good. You've got it.
It's not in the hole.
Yeah, it's lined up though. Get in there. Woohoo!
All right.
Yay.
Nice.
Redemption.
Thank you.
All right, so that is push core 5.
That was NA12446 push core 5.
Pretty shallow.
My mom just asked, she's watching. Hi, Mom. What are the white things? Those are brittle stars. Did we end up finding a genus that we think those are? Or a
Maybe
species?
Opium Museum, did we decide? Very common down here.
I think we're still working on that. If Steve's in the chat, though, maybe he knows.
But we know it's a brittle star, maybe just not exactly the type. And they seem to be loving this high food, higher oxygen sedimented region that we've landed on. Maybe they're feeding on some of the food that and the microbes that the microbiologists on this team will find in the push cores that we're collecting right now. That was PC4?
That was PC4, yes.
169.
169.
Thank you. Yeah, sorry to everybody tuning in. We tend to get a little quiet during the coring as we excitedly watch the ROV pilot lift them out of the sediment and carefully put them in the
Especially when he botches it.
It's no easy task.
You don't want to make it look too easy, Bob.
The two back ones are the hard ones.
Yeah.
It's easier from there.
Front.
Do you prefer us to be quiet, or is that too much
I
pressure?
don't care. When you're quiet, I just know you're unhappy, so.
All right, we'll keep talking then.
It's respectful silence, not unhappy silence.
We don't want to interrupt your focus.
We think we're trying to let you concentrate.
It reminds me, I grew up playing volleyball, and there was always this moment before the serve, and I could also never decide if people were talking or yelling, it could be really distracting, but if it got totally silent, everyone's just staring at you.
I guess you've gotten really good at tuning us out, probably, when you need to.
No, the worst if you ask a question and you get silence back, that's when you know you're.
All right, push core 3, that was 170.
Correct, 170.
Not very deep.
Yeah, surprisingly shallow.
I think they go all the way in, but then the sediment is soft and must create some sort of suction that pulls them out again, pulls it down. I think they'll be fine. You need 10 to 12 centimeters, right? So they should be okay.
Yeah, we should be okay.
So I was watching you guys a little bit coring the other night. So you kind of slice them out in discs. Is that how you're doing it?
Yes, correct. Into 1 centimeter thick discs of sediment. And we kind of like put them into sterile plates. Oops. And then